My name is Jonathan Choquette. I'm the lead biologist on the Ojibwe Prairie Reptile Recovery Program with Wildlife Preservation Canada. I'm also conducting my PhD part-time with Laurentian University under the guise of the OPREC program. So OPREC is Ojibwe Prairie Reptile Recovery. I'm going to use that acronym a little bit tonight. And I thank you all for coming tonight to listen to me speak about Massasauga recovery at Ojibwe Prairie. So I'll talk to you for between half hour and 45 minutes tonight. I'm going to give my talk in 3 parts. I'm going to open up by giving you a little bit of a background on the Massasauga and on the OPREC program. And then I'm going to jump into a review of some of the work we've done over the last 5 years. And then I'm going to dip into some of the PhD work that I'm working on as part of the OPREC program. So without further ado, let's get into part 1, Intro to Massasaugas and OPREC. So I know we're in the middle of a global pandemic, but let's not forget that we're also in the midst of a biodiversity crisis. Barnowski et al. In 2011 asked the question, has the Earth's 6th mass extinction already arrived? And they basically say that there's an urgency to relieve the pressures that are pushing today's species towards extinction. And reptiles have not escaped this decline. And back in 2000, Gibbons et al. Stated that reptile species are declining on a global scale. And we know that here in Canada, at least some of us know, it's not really a common fact that most of our reptiles are at risk. And actually, reptiles are one of the most endangered group of vertebrates in our country. And that all trickles down to the local level. Here where I work in Essex County, we found just recently that the majority of reptiles and amphibians that occurred here historically are either gone, extirpated, extinct locally, or they are extremely rare. And one of those species is actually the Massasauga. Massasaugas are interesting in the fact that they are the only snake in Canada that's also not just endangered in Canada, but is of global conservation concern, meaning it's declining across its global range. You can see the global map here of Massasaugas runs from, the range goes from central Ontario all the way down to southern Illinois. You can see all the counties where the animal has become extirpated in the red X, and there's Ojibwe Prairie right smack in the middle in Essex County. And the Ojibwe Prairie is unique because our population of Massasaugas here is the only one in Canada that lives in a tall grass prairie. You can see the beautiful landscape in the photo there by Russ Jones. We are isolated from the rest of our Canadian counterparts. The closest population you'll see in the map there is Wainfleet Bog, about 300 plus kilometers to the east. And it's only one of 2 Carolinian populations left. We know of approximately 17 that were documented historically. There's only 2 left, Ojibwe Prairie being one of those. It's actually more similar to its American counterparts across the river in Michigan, and genetically is unique. It's actually more similar as well, not only in the prairie habitat, but also genetically to its American counterparts. And the one thing about the Ojibwe Prairie is it persists in an urbanizing landscape of West Windsor and the town of LaSalle. You can see in the map here, it's surrounded by development and roads, and that's really put a number of pressures on this population. A history of road mortality, of intentional killing by people, and of residential development have resulted in addition to a number of other threats, particularly urban related threats, have resulted in a history of decline here, precipitous decline since the 70s, 98%. These animals have really come to the point of blinking out, and they will become extirpated if we don't intervene, essentially. And it's important that we do intervene because these animals have significance. They have cultural and community significance. They're protected by a number of laws going from the provincial all the way up, going from the municipal all the way up to the national level. And as I mentioned before, they hold a unique component of not only Canada's biodiversity, but of global biodiversity. And the thing here is that they persist in a globally rare habitat. So when we protect Massasaugas and we protect their habitat, we protect a very unique ecosystem, the tallgrass prairie. And the Ojibwe Prairie is often cited as the largest protected tallgrass prairie in Ontario, one of the largest. So really important that we protect and recover this population. And so this is where Wildlife Preservation Canada comes in. So who are we? We're essentially a Canadian organization that focuses on saving animals that are facing extinction. And we do that really by specializing in captive breeding and conservation translocation. So really the hands-on techniques to bring populations back from the brink. We're based in Guelph. We've been operating since 1985. We do projects from coast to coast. And we also help to build Canada's conservation capacity, not only through collaboration, because collaboration is key in a number of our projects, but also through helping to train young scientists. And I like to point out this quote. It kind of sums up what we do here. Conservation is more than preventing absence. It's also about creating presence. So let's talk a little bit about OPREC. And what is OPREC? Well, essentially, it's a program that's been administered by WPC since 2013. Before I was employed by WPC, I was self-employed, and I partnered with them to start this project and to help secure funding through Ontario and Canada government grants. This program is multifaceted in the sense that we're targeting the Ojibwe population of Massasaugas. That's our real goal, is to recover the Ojibwe Massasaugas, but we're also designing our work and doing our work so that we can benefit a number of other species at risk, reptiles and plants that live in the tallgrass prairie. So, and there's lots of different components to the recovery program, which I'll get into. But what we're doing is we're taking the recovery strategy, which is a federal government document, which recommends what needs to be done to recover the species. And we're really taking the key points that are recommended for the Ojibwe prairie population, and we're implementing those recommendations on the ground. So it's really targeted implementation of the recovery strategy. And that was released draft in 2013, and we've been following that guidance ever since. I mentioned our major funding, and basically half and half split. Most of our fundings come from the provincial government on one side and the federal government on the other side through some of their key endangered species and habitat programs. And this is, I mean, we wouldn't have been able to do what we've done thus far without help from the provincial and federal government. So a huge help to them. We've been fairly lucky to be funded most years since we've started. So this has been great. The other important thing is that we've had a number of key partners which has helped guide this recovery work. Since 2015, we put together a working group, the OPREC working group, and this is a volunteer group of stakeholders and land managers that essentially comes together. We meet about 4 times a year to advise on recovery and guide recovery projects, as well as to develop implementation plans. So that's essentially a plan to implement the reintroduction and the recovery of this population. And we come together under this umbrella or this vision of a future where the Ojibwe population of Massasaugas is of a healthy, viable size and distributed across a connected park system, and where threats are abated and where human-snake conflict is properly managed. And you can see the key members of the working group are represented there by their icons, by their logos. This recovery strategy is what I've been talking about. It guides recovery of not only the Ojibwe population, but Massasaugas in Canada. But recovery looks like a number of key things, everything from maintaining the habitat all the way down through augmenting the population through reintroduction. So it's these key recommendations that we are taking and building into our recovery program. It's also important to note that our work is really focused in on what the federal government identified as critical habitat. You can see it circled in red here. And this critical habitat spans 2 municipalities, Windsor and LaSalle. We have to work with a couple of different regulators to get permits to work on the lands in that within the critical habitat. And there's 4 major property owners that we work with. So that's where most of our work is happening, in that critical habitat. So now that we have a good understanding of the background of our target species and of the OPREC program, I will discuss our progress over the last 5 years and give you a good sense of some of the different and diverse types of activities that we've been doing. First thing is population monitoring. So an important part of recovery is understanding your target species. And we invest over 1,000 person hours every year monitoring the population, assessing threats, and surveilling the habitat. And this is all important because all this information helps guide the work that we do and helps us to manage adaptively and adjust what works and what doesn't work on the fly, essentially. Another important thing that we do is enhancing habitat. And habitat becomes degraded. We have succession by shrubs. We have invasive species. And so a couple of important things that we do. Every year, actually, we enhance habitats within about 5.8 hectares. So in 5.8 hectare plots, we're enhancing habitats, and we're doing that through the building of woody debris structures like what you see here. We've built over 150 of these brush piles and log piles to help Massasaugas. And we also have been removing over 775 kilograms of invasive plants to help promote the tallgrass prairie habitat and the endangered plants that live therein. And through our monitoring, we've actually been able to determine that those woody debris features that we're are quite effective. Massasaugas give birth to live young. Males will choose like a log pile like this and will gestate there until she gives birth to live young. And we've identified, we've observed an average of 11 observations of species at-risk snakes using our woody debris features every year. So every year we've had Massasaugas or fox snakes using these features. And in 4 of the last 5 years, we've had 1 or 2 gravid females using our enhancement structures and even some babies born at them. So really effective, something that we're going to continue to perfect and to do or in the coming years. Now, this is unfortunate. Many of you see dead animals on the road. This is a fox snake, and this is a snapping turtle. And roe mortality, it exists in this landscape. It's a threat to our species at risk. And so we want to know where it's happening and what it's affecting and how often it's happening. And so over the last 5 years, we've actually recorded over 5,500 road-killed vertebrates on roads that bisect the Ojibwe Prairie Complex. And we use this data to identify hotspots. Where are the animals getting killed in greater numbers, and where can we put up fencing like this to keep them off the road? So we've installed an average of 287 meters of barrier fencing every year since 2015. Most of that's this temporary black stuff that we put up during the active season, we take down at the end of the year, but we've actually started getting into some permanent fencing. You'll see in the bottom right here where we put up 260 meters of permanent snake mesh to keep rattlesnakes out of residential backyards, keep people safe, keep snakes safe. And we're moving more into this permanent stuff. We've actually just installed 300 meters this year to keep snakes off the road. Habitat protection is really important. And in terms of habitat protection, one of the key things that we do is awareness of the importance of habitat and submission of records to provincial databases. So for the first part, in 2016, we partnered with Lakehead to actually identify the Ojibwe Prairie Complex and Greater Park ecosystem seen here. And then we nominated it to the King Herpetological Society and to hopefully get it designated as an important amphibian and reptile area. And we were successful. In 2016, it was, I think, the 11th in parasite in Canada. This is similar to the important bird areas. It doesn't give legal protection, but it essentially helps identify the most important areas in Canada for our reptiles and amphibians. So we got a lot of media attention here, and it was great. And this is basically the landscape and the designation that we work with moving forward. And we're always working with partners to try to increase habitat protection within this landscape. And we also, within that area, we submit our endangered species observations. And that's an important part of habitat protection because the province doesn't have the observation data. They can't protect the habitat. So we've submitted over 1,800 species at risk observations in the last 5 years. That's species at risk reptiles and species at risk plants that you can see on the right. Really important that we submit those observations because if we don't know that the animals are there, then the habitat really can't be protected. So that's a key part of habitat protection. I also mentioned that the Ojibwe Prairie Complex is fragmented. So it's the main parks that we have protected, under protection, are subdivided from each other by roads, by residential areas. And so one of the things that we've been doing is to try to identify corridors. And here's a map here of a 5-kilometer long, 17-hectare wildlife corridor that links habitat. And we've been working, this is a right-of-way. This is a utility right-of-way. And so we've been working with Infrastructure Ontario and Hydro One to allow us to manage habitat for tallgrass prairie. And our main goal here is to create a 5-kilometer long tallgrass prairie corridor that goes right through the spine of the Ojibwe Prairie Complex. So we're working on different components, making brush piles in here, working with partners to install eco passages where the corridor intersects roads. This is basically guiding us in how we move forward with our partners to connect these habitats. We're working in an urban landscape. We're working with the venomous species. We need public buy-in to recover Massasaugas. There's no doubt about that. And so public outreach is a key component of what we do. And that includes things like installing signs. We've installed 15 public outreach signs in the last 5 years, everything from interpretive signs seen here with the mayor of LaSalle to driver awareness signs at road mortality hotspots, to Massasauga information signs in the park so people know, not to just pick up any snakes willy-nilly so they don't get bit. And we work with homeowners and park users who use the parks and who live around the parks and the critical habitat parks. And we've delivered over 2,000 outreach materials to residents. We've also done questionnaires so that we can better understand if our outreach is working. And we're using that information to help better fine-tune the messaging that we're delivering to people. But we have a pretty good sense of the support for recovery, and it's quite high. It's highest right around the provincial park. So that's really good to know for moving forward with reintroductions. Another part of public outreach is giving talks like this one, sharing information about the recovery work, sharing information about Massasaugas, the importance of the tallgrass prairie. We've given 19 public presentations since 2015. We like to get our work in the media. We've been featured in over 15 media stories, mostly local media, but some provincial, a little bit national, and 2 of our official languages. So that's really important too, to get the word out there. And I don't want to forget about population augmentation, because the population that we're working with is so small that it will not be recovered. It will not persist unless we restock the population with new animals. We do have a captive population spread across a number of zoos. Toronto Zoo, Detroit Zoo have Massasaugas, and they manage them as one and trade amongst each other. However, there aren't enough animals in the captive zoo to produce young for reintroductions. So we've been working with a bunch of different partners. You see a list up here. Some of them are still working with us, some of them aren't, to try to find out how we can increase the captive population size. And we've been working since 2015 on this. We actually just got some great progress this year. We got the permits in place and through partnerships. So we have 14 new snakes collected from healthy populations up in central Ontario to add to that captive population. Our target was 30. We're already halfway there. So that's some great news. So that's a quick rundown of some of the work that we've done in the last 5 years. And now we're going to move into my PhD research, which is happening under the umbrella of the OPREC program. And the main topic of my research is reintroduction biology. So reintroduction biology is the study and practice of trying to establish populations using translocations. So translocations are the movement of an animal or group of animals from one place to another for the purpose, in this case, of conservation, for the purpose of reestablishing populations. The problem is we really, for a number of different animals, we don't know how to make translocations work. We don't really know how to establish new populations. And snakes, I mean, particularly for snakes, there's a lot of unanswered questions. We still don't know, when is a site good enough? When have we addressed all the threats to reestablish a population? All the way to, like how do we overcome the challenge of overwinter survival in temperate zone snakes? There's a number of questions that have been put forth in 2009. And even more recently in 2014, when it comes to translocations of herpetofauna, which are reptiles and amphibians, we're still dealing with how to make the techniques better. Germano et al. Said, technique improvement is a dominant theme. Captive husbandry and release techniques, all the way through to evaluating appropriate methods. We're still trying to figure these things out to make translocations work. And habitat quality is still one of the greatest reasons for translocation failure. But we have our federal Massasauga recovery strategy, which essentially suggests or dictates or guides that we figure out if translocations are effective to manage populations of Massasaugas. We should determine the feasibility of augmenting populations using translocations to increase population size or distribution. So this is the guidance that we're given, and the language that we have in order to move forward with determining if translocations will work for Massasaugas, which brings us into my PhD, which is about improving not only captive breeding, but conservation translocation techniques to recover temperate zone rattlesnake, the Massasauga. So I'm conducting this PhD work part-time at Laurentian University, and my advisor is Dr. Jackie Litskis, and I'm being co-advised by Dr. Trevor Pitcher down here in Windsor at the University of Windsor. And Renew Zoo is an important funder of this work, and this is an NSERC program that links conservation practitioners like myself with zoos to really help foster conservation work in zoos. So I'm going to go over the 5 chapters of my PhD, all of which are related to Massasauga recovery. I'm going to skim over the 2 last ones, but I'll go into a bit more detail of the first 3 ones because we've actually collected some data, and I can talk to you a little bit about some preliminary results. So the first chapter is all about reviewing correlates of translocation success with snakes. So what works and what doesn't work for snake translocations. And this is important because we're going to talk about this document. You're going to see this document a lot here. This is the guidelines for reintroductions and other conservation translocations that's put forward by the IUCN. The IUCN is the International Union for the Conservation of Nature. So they basically say that we need to figure out the most appropriate life stages for translocations before we do them. We need to figure out the correlates between pre-release behavior and post-release survival. So, what is it about raising animals in captivity that results in whether they live or die once you release them? And they acknowledge that low survival once you release an animal, it can be due to a wide range of factors. So there's, we need to figure out a number of things here that are consistent with the research questions that remain for snakes. And we do have some guidelines, because I point out here a number of review papers that have been published that talk about what works and what doesn't work for reptiles and amphibians in general. And this gives us some good guidelines, no doubt. However, snake translocation projects specifically have only ever made up a small percentage of papers reviewed in these studies, from 4% of 33% of projects included in these studies. So we really don't have some snake-specific guidelines, and there hasn't been a review out there specific to snakes. And when it comes to Massasaugas, which is our target species, we do have some guidelines, or some guidance rather, from a handful of Massasauga translocations that have been conducted. And we do know that generally the animals seem to have a fairly normal survival rate in the active season. It's winter time that really kills them off. And this paper here shows a study that was done at the Ojibwe Prairie back in 2006. And 27 snakes were released by Karen Cedar, who works at the Ojibwe Nature Center, and Andrew Lentini is an author here. He's at the Toronto Zoo. And unfortunately, none of those animals survived the winter. So there's still some real unknowns in how to make these translocations work for Massasauga. So I want to find out what works and what doesn't work for translocations, which techniques are associated with survival of snakes, both in the short term and then long term, and when it comes to population establishment, for example. And to do this, I want to do a systematic and non-systematic review of the literature, and not only the published literature, but also the other unpublished literature out there that talks about snake translocations. And so thus far, we've assembled 109 unique sources, so published papers, book chapters, etc., that talk about a translocation that was done and also present some results. And these information sources represent 118 different case studies spanning 41 different species. So we have a lot of information now. Now the next step is to analyze this information and look at which factors are associated with success and which aren't. And actually the 3 species, the photos on the bottom here, the Massasauga, the timber rattlesnake, and the common water snake, these actually make up 30% of all of those case studies. So these species are quite commonly translocated and quite commonly published on. So we'll get some good information on those species as well. And ideally, in the end, the significance of this chapter is to really synthesize the snake translocation research and provide guidance on the most likely factors to influence success and hopefully improve reintroductions of snakes in the future. And this is just a diagram of showing what the results will look like in terms of the factors on the y-axis, and essentially the results on the x-axis here that would give us an image of which factors are more associated with successful translocations and which are not. And then we move into chapter 2, which is all about evaluating appropriate monitoring methods. Back to this IUCN guidelines. They recommend that we collect baseline information before conducting a translocation. Makes sense, right? I mean, if we don't know much about the population that we're trying to reintroduce, I mean, it's really difficult to ascribe the changes that we observe with the actual impact of our releases, particularly when there's still animals alive on the landscape. The problem with snakes is that they're really hard to find, and rattlesnakes are really hard to find. And so, you know, we're dealing with trying to estimate the range, the distribution of a cryptic animal, really hard to find animal. And so occupancy modeling has become more and more popular as a way to do that, because it incorporates the fact, what's called imperfect detection, meaning that every time you go and survey for the animal, you don't find it. And so it builds that into the analysis. And you look at DP here, DP means detection probability. And so when detection probability is less than 1, that means that you're not finding it every time. 1 meaning you find the animal every time you go. With Massasaugas, that's not the case. You have to survey many, many times before you find the animal. And so this analysis, I want to use this to evaluate population establishment of Massasaugas and determine whether or not this is a useful approach to evaluating our reintroductions in the future. But there's some caveats here. And the thing is that with any modeling, there's some basic assumptions that we need to respect. And if our detection probability is too low, so DP less than 0.5, basically we have unreliable results. So in those cases, it might not be a useful approach. And you know, finding a snake out in the wild is difficult in and of itself, but it's also, the likelihood of finding a snake is influenced by a number of factors. Whether the species is highly abundant, what season it is. So it's really important that we determine, you know, we standardize how we're searching for this animal, because that will affect detection probability. So essentially what I want to do in this chapter is to estimate detection probability and occupancy being the range of Massasaugas at the Ojibwe Prairie before we even do any translocation. So really understand the state of the population and the likelihood, or which techniques will respect model assumptions for occupancy modeling and maximize that detection probability. So all this work has been done. We conducted a number of repeated surveys, standardized surveys, using published protocols and testing out different methods. We looked at spring versus summer surveys, single surveys versus team surveys, and 2 different methods. You'll see this VE is visual encounter, and that's just looking out in the habitat for the animals. ACO is artificial cover object, and that's putting down wood boards or tins and looking for snakes underneath. And then we analyzed this data using an occupancy modeling program called Presence. So we have 6 years of data, over 3,800 or approximately 3,800 person hours of surveys for these animals out of Ojibwe Prairie. We have over 220 unique observations at only 4 sites out of our 40 though, so the range isn't very big. But we started doing some analysis with this data, and unfortunately, the ACOs, the cover objects, were not a successful method at finding Massasaugas. They were successful at finding 5 other snake species, but not a useful approach for surveying for Massasaugas. Visual encounter surveys, on the other hand, are effective, but we have low detection probability. So you can see detection probability in the y-axis ranging from 0 to 0.6. And then you can see the year of study in the bottom axis right here. And although you can see the first year we had a pretty high, relatively high detection probability. But by the way, 0.3 or 0.4 means every 4 surveys out of 10 we find the animal. Okay, so here, unfortunately, when we have high detection probabilities, generally because you have a pregnant female being seen day in, day out at the same spot, because they tend to hang around at one spot, and that artificially inflates detection probability. That's not good. We can't really, it violates our model assumptions. But group surveys in the spring are the most promising. And so this is where we have this is basically where we need to guide our monitoring methods in the future in order to use this method to evaluate our translocations. So hopefully these results will not only give us baseline data pre-translocation, will also help us to improve techniques to evaluate translocations for Massasaugas. And the results here have really pointed out the importance of trialing different methods before you do your translocation, so you can really be confident in how you're going to measure success. Which brings me to chapter 3. Again, before we do any translocations, and because habitat is so important, we need to select and validate where we're going to release the animals. And again, Germano et al., we saw this already. They said that habitat quality or lack of specific habitat characteristics are one of the greatest reasons for translocation failure. The greatest reason. That's for herpetofauna. And the IUCN here in their guidelines also point out that it's essential to evaluate the current suitability of habitat in any proposed destination area. So I'm listening, I'm not going to ignore all that advice. In addition, I'm cognizant of the previous reintroduction attempt that happened in 2016, where none of the released, 2006, where none of the released Massasaugas were alive following the winter. And these authors, they state specifically that repatriation is a synonym for reintroductions. So reintroductions will only be successful if snakes are released in close proximity to their hibernation sites. That's because they found that the animals would actually come back to near the area where they were released to try to find a hibernation site. So the idea is that we should release them where there's good hibernation habitat, under the assumption that they will come back to try to hibernate there. So I wanted to evaluate, first identify, and then evaluate suitable release sites for Massasaugas based on hibernation habitat quality. And essentially, I want to know what the overwinter survival rate of snakes artificially hibernated at those sites are. And specifically through the help of a colleague, Ann Yagi at Wainfleet Bog, I identified 4 1-hectare grids in the middle of the nature reserve. You can see them in the box there. They've been outfitted with groundwater wells and frost tubes, and those have been monitored since 2015, so we could figure out the space underground called the life zone. Where does it remain frost-free, below the frost line, but above the water table for the whole winter, so that any animals in there hibernating underground would survive? So I use that information to identify release sites, 4 release sites within those 1-hectare grids that you see in that red square. And then, this is pretty cool. And then at those release sites, we installed 10 of these artificial hibernacula. So these were designed to mimic crayfish burrows, which are the natural hibernacula used by Massasaugas. And with some advice from, again, Ann Yagi and some colleagues in Hungary, I designed this artificial hibernacula, and we installed them. And then last year, we intentionally hibernated 21 eastern garter snakes. You can see those guys in the picture down there. And you can see the hibernacula when it's installed next to the garter snakes. That's what it looks like. It's installed in the ground. So we hibernated 21 eastern garter snakes. That was just this last winter. And I'm happy to say that everything went super good. The air temperature in these hibernacula was quite stable. The humidity levels were high, which is what we're looking for. You can see some pictures of the snakes in the tubes, and we evaluated them through a borescope every 2 weeks. And in the spring of this year, almost all of those snakes survived. We had only 1 snake die over the wintertime, which is really promising and suggests that the method we use to identify hibernation sites and ergo release sites is actually pretty good. So we're going to study these sites for a 2nd year to increase confidence in our results, and then we're going to go from there. But essentially, this study will help support a rigorous release site selection method, and ideally help myself and other practitioners to overcome that significant barrier to translocations, which is the overwinter survival. There's a nice article put out by Canadian Geographic in June of this year that talks a little bit about this research. So if you guys are interested, you can go and check that out on Canadian Geographic's website. Finally, I'm going to briefly talk to you about chapter 4 and 5, more or less together. These are the captive breeding side of things. So in chapter 4, I want to help improve captive breeding protocols for Massasauga. And then in chapter 5, I want to help develop a sustainable harvest and release strategy. So give you a brief outline of those. So I talked to you about the fact that there is a captive population of Massasaugas, and it's managed by the Association of Zoos and Aquariums. Now, in these two documents, the AZA here state that we need research into the environmental and physiological requirements for inducing reproductive behavior in Massasaugas. So essentially, we need to find ways to increase reproduction of these animals in captivity. And in this other document, they go out to say that we want to develop standard reproductive protocols. Okay, so those are basically what's put out there as important for the captive population of Massasaugas. So what I want to do in chapter 4 is I want to figure out which conditions in the zoos have resulted in greater reproductive success. And then I want to use that information to help develop, or recommend rather, standard reproductive protocols. And I'm going to do this by collecting and analyzing data from about 27 captive adult female Massasaugas that are known right now in the captive population. And the importance of this is that ideally it would contribute to, the recommendations would contribute to increasing the number of captive bred animals that would be available to do translocations at not only Ojibwe Prairie, but elsewhere across the range of this species. Now, talking about chapter 5, I'm pointing to that reintroduction guidelines again. And essentially, the IUCN state that modeling should be used to predict the outcome of a translocation under various scenarios as a valuable insight for selecting the optimal strategy. And then Canessa et al. In this paper down here state that releases deplete the captive population and can reduce its viability, generating a trade-off between the insurance and reintroduction objectives. So in other words, we need to figure out how many animals to release every year and for how many years. And if we're harvesting from a captive population, we need to know how many animals can we harvest from that captive population and still sustain that population. So some important questions when we're planning a reintroduction. So what I want to do is use this thing called PVA, Population Viability Analysis. And it's a modeling approach to compare competing reintroduction scenarios affecting wild and captive populations. Okay, so essentially I would assemble demographic data like birth and death rates and age of maturity from the wild population, so that's from the Ojibwe population or nearby populations. And with demographic, I also want to assemble demographic information from the captive zoo populations and then analyze that in this program called Vortex. And Vortex is a program that does PVAs. And ideally, the outcome here, the significance here, is to provide annual release targets that would help establish a wild population while sustaining a captive wild source, right? So essentially it would help us understand, given the growth rate in the captive population and their target population size in the wild, how many animals can we take from the captive population every year and keep building it and build the wild population? So it's quite complex, so that's why this kind of analysis would be very helpful. So let's bring it all home. Conclusions. So what did we hear today? Well, we heard that Massasaugas are globally imperiled and the only snake in Canada that's of global conservation concern. And it's in decline. It's in decline across its range, and it's really in decline at the Ojibwe Prairie. This population needs hands-on recovery techniques or it's gone, is essentially the point here. And so the OPREC program, the Ojibwe Prairie Reptile Recovery Program, has really been implementing the recovery strategy since 2013. And we've had some successes in habitat enhancement and mitigating threats and building partnerships, etc. But there's still more work to do to prepare that landscape and to get ready for successful translocations. And then we heard about the PhD research that's going on, and this is important because we know that we need to improve conservation translocation techniques for temperate zone snakes. And that's what the PhD will aim to do, to try to get some better techniques out there that not only we can use, but that other practitioners can use. Because when it comes down to it, effective reintroduction techniques are really a key component of biodiversity preservation. So I want to make sure that I thank the major funders and the supporters that have been helping us develop OPREC and helping us to recover this population of Massasaugas. And I thank you all for listening, and I'm happy to answer whatever questions that you have. Thank you very much.